drive us toward wellness and prevention and ultimately an N-of-1 medicine, which is really where we want to go. And I'd say with data-driven health, there are really 4 key features that you have to keep in mind. So one is the idea of big data. I'll define what that is precisely. The other is the idea of you have to use systems approaches to analyze that data and deal with its complexity. And the third is humans are unbelievably complicated, and the conventional assays we have for deciphering this complexity are just beginning to scratch the surface. And I'll show you some whole new types of technologies that give us different views of data-driven health. And then finally, I'll make the point that AI is going to be transformational for medicine in 1, its ability to classify information, and 2, its ability to deliver it effectively to physicians and patients. And we'll talk about how we proceed with that. So I've had 60 years of a science career, so let me give you a framework for how I think about data science that starts when I was a young professor at Caltech in 1970. I met Harold first back in those days. And my major interest was human complexity and the realization it's unbelievably difficult. And what was obvious to me at that time were 2 things. 1, the only way you're going to deal with complexity is big data. We have to be able to look at all of its dimensions and so forth. And 2, it was clear we really needed a way to think about how to analyze that data. And that later became systems biology. So I've lived through a series of paradigm changes that I'll run through, and they really have framed my whole view of data-driven health. So the first at Caltech, I developed a lab that had enormous technological capacity, and over the years we developed 6 instruments: DNA sequencers and synthesizers and protein sequencers and synthesizers. And one that plays a really key role in the very last slide is an inkjet technology for the rapid solid state of enormous numbers of DNA sequences. So that was the framework for bringing big data into biology. And of course, I got invited to the first meeting ever held on the Human Genome Project in the year 1985 with 11 others to opine on whether this was a good idea or not. And we came to 2 strong conclusions. 1, it was technically feasible but difficult. And 2, most people hated it because it was big science and it would threaten science. And of course, what finishing the genome did was really marvelous from the point of view of health, because the genome sets the framework of potential for each of our health trajectories. And I'll talk more about that later on. The Caltech faculty were really angry that I had a big lab, and I got severely criticized. So I went to them and said, look, let me start a department where each technology is a faculty member, cross-disciplinary department. Biology turned me down class, so Bill Gates made it possible to go to the University of Washington and start such a department. And in its 8 years of existence, Phil Green developed the 2 key pieces of software for the Human Genome Project: assembly and quality assessment. Number 2, John Yates and Rudy Abersol pioneered the first 2 really key technologies in proteomics and led it for a lot of the rest of the time. And 3, we developed this inkjet technology, which is still used by Agilent to do large-scale DNA synthesis and so forth. But what happened is I have actually an anathema to big bureaucratic organizations. So after 8 years there, I resigned and started the Institute for Systems Biology, because I really wanted to do that at the University of Washington. And bureaucratic complexity really made it impossible. I could give you 10 examples of why this is so. But systems biology then was really an exciting idea. And one of the first projects we did with Trey Eidiger was to look at glucose utilization in yeast. We mapped it into dynamic networks. We could make predictions about all sorts of things. And it was clear this was the way to go forward and so forth. And then in the early 2000s, we came up with this idea of P4 medicine, predictive, preventive, personalized, and participatory. And I will say the first 3 Ps are science. And we basically know how to do those now. It's just a matter of scaling things. The 4th P, participatory, how you persuade patients, physicians, healthcare leaders, et cetera, to exchange a healthcare of wellness and prevention for one they make all their money on now, disease-driven healthcare, is an enormous economic, educational, sociologic, and psychological challenge. And we could spend 20 minutes talking about how we've approached that. But rather, what I will say is, in the context of doing P4 analyses, we did a number of really interesting things. For example, we were able to look at 25 different organs in the human body and identify organ-specific proteins in the blood. And what that gave us the ability to do was to test in those organs state changes. And we did a bunch of things, but one of the most interesting one is we compared the difference between a normal patient and amphetamine poisoning in mice. And we mapped out, we had 44 organ-specific proteins, of which 27 were perturbed. They mapped into networks. You could see the kind of thing that was happening. And then we did the same thing in amphetamine poisoning in humans and showed enormous homology in the networks and the dynamics and so forth. So that is going to be a really important approach for looking at drug toxicities in the future. And we'll come back to that point again. The second one, the first disease we looked at with regard to a systems approach was really prion disease in mice. And those results in this slide are absolutely spectacular. You could induce the disease at will by the infection with the disease prion particle. And what it did in neuropathological terms was go through 4 stages. Prion accumulation and replication, glial and astrocyte activation, synaptic degeneration, and then neuronal cell death. And we were able to map networks that precisely defined each of those features in really quite a remarkable way. And the important thing about that mapping was the disease-perturbed networks increased exponentially. So when you got to the final disease, it was impossible to think about therapies. But at the beginning, there were simple perturbations that gave us insights into what the disease might be, and you could think about therapies. We didn't follow that up. But the final point that was really important is that each of those stages, the mice secreted brain-specific blood proteins into the blood that showed differences that marked the onset of each of those 4 stages. And what this suggested, and we followed it up later in humans, is we can use these kinds of analyses to actually get very early stages of detection. And my major point in this lecture is the future of chronic disease is all about early detection and early therapeutic reversal. And I'll show you just how we propose to do that. So with that in mind, what let me just say, we did exactly the same thing with glioblastoma in mice, and it was precisely the same kind of facts and everything. It was really quite compelling again. And what happened is we generated enormous numbers of strategies and potential new technologies, which I really had trouble getting funded at NIH. And so what I did is I went to 4 different countries and tried to argue, we'll set up a systems biology institute for you if you'll help in a collaborative, supportive kind of role. So I was 0 for 4 on the first one. It's a great story. Every one was for different reasons, but the 5th one, where the chair of my board said, I'll believe in the tooth fairy if you succeed this time, was Luxembourg. And what happened is I had the good fortune to meet the Minister of Economy, who at that time was the real driver in government there. And he was determined to bring biotech and health to Luxembourg. So he said, well, Lee, I'm really convinced by this systems biology stuff. Write me a proposal. So I said, what are the limits? And he said, there are no limits. You can ask for what you want. So I asked for 100 million, and there was no haggling. We got 100 million for 5 years, and it absolutely transformed what we could do. And we built up a center at Luxembourg that is one of the leading biosystems centers again. And just recently, we've closed the loop, and we'll talk about that again later on. But what happened in 13 then with all these new technologies and our ability to recruit some really exceptional people from the Luxembourg money was that I put together a proposal to persuade 108 of my friends to go through a year-long study where we did genome and phenome analyses and saw what we could get out of it. And I remember when Francis Collins in 13 saying, look, Francis, I've got this terrific wellness project. And he said, the role of NIH is to study disease. We have no interest in wellness. So we raised it from private money and elsewhere, and we did this small project. It was spectacular. We set a wellness company called Airedale, where we recruited 5,000 people over a 4-year period. And this is the data that actually sets the framework for what you can do with phenomics now. And we're going to go through all the different things you can do. 2 more paradigm changes. One was when I started as chief science officer at Providence with the duty to bring genomics and scientific wellness to Providence. After 5 years, I decided big bureaucratic hospital systems are even worse than big state universities. So I started this nonprofit called Phenome Health, which we've used since 21 to drive this whole idea of data-driven health. And now we're at the point where it's clear what we need is a 1 million-person project, and I'll tell you what it is and why it's so important in just a moment. Now, I was going to talk about a new AI-driven, information-based peptide drug company, but I don't have enough time in a half hour to do that. So I'll mention a word or two of it at the end. But my prediction is this company will transform the pharma industry in the next 10 to 15 years, and I'm glad to show anybody who's interested just exactly what we're doing. We're doing this with 3 people from Sweden and 3 people from the US, including Ingemar, who's here. He can answer questions too. The key points I would make, 7 of the 8 of these so-called paradigm changes were initiated by small, agile organizations. Some were nonprofit and some were for-profit, but my belief this change comes through small organizations that have agility and can make a complete commitment to a vision in a way the big companies of the world or the big academic organizations of the world can never do. The data-driven health, of course, is all about the genome, and we determine the entire genome. Let me just say that the first genome project now, with all that's gone on, has brought the cost of sequencing down by 7 orders of magnitude, and you can get a genome for about $100 now, not its interpretation, but a genome. And what, of course, the genome did is set the framework for what you can do as you go from infancy to death. It's the scope of your potential. And what influences within that large envelope that you have are 2 really major things. So one is your behavior, and the other is your environment. And I read a recent paper that said, if you talk about health, 11% is genome, 89% is phenome. And it's true. We could talk a lot of reasons why I think that. So, and the phenomics, of course, is taking blood measurements and gut microbiome and digital health measurements that let you assess that change in trajectory across your phenotype. And of course, this million-person project is necessary because it will be the ultimate validation of the power of this approach to do 3 things. 1, enormously improve individual health. 2, leave very large cost savings from getting rid of most chronic diseases. And 3, catalyzing the technology that can take these very complicated multi-omic signals and transfer them into wearables that can be done at home. And that's the democratization you need to be able to think about going to underdeveloped countries with this thing. And we're well on our way to being able to do these things, as I'll show you. The big data analysis, of course, was first the idea, because blood bathes all your organs and they secrete molecules, you can quantify those molecules and you can see state changes in organs or in the system as a whole. And that, of course, made blood a window into wellness and disease. And what we need are about 5 more major diverse windows into health and disease. And I'll talk about one more we're exploring right now later on. So the idea is, what do you do with all this digital data? And of course, there are 2 approaches now. You can do dynamic biological networks, map data into them, or biological communities. And I'll explain the difference. It's a really important difference, those things that you understand, because they have to do with very different dimensionalities of information. And then, of course, there are AI things that are centered on knowledge graphs and digital twins and transformers, large language models, and things like that. And I'll show you how we're going to use those things. With the Airedale data, a 4-year program, we have 6 data features: genome variants, 3 analytes in the blood, proteins, metabolites, clinical chemistry, the gut microbiome, and physiologic measurements with the Peptide. And what was really key in this was we had coaches that both delivered actionable possibilities, the things that derived from the data that patients could then do to improve wellness or avoid disease. And what is really interesting is coaches are not scalable. If you're talking about doing 100 million patients, you need a coach at best for 1 in 1,000 patients. So chatbots are going to be our coaches of the future, and I'll talk about how we're doing that. We have almost 30 papers now that have come out of these data, and each of them gives you a new view of scientific wellness and prevention and deep insights. And I'm going to summarize all of these things in one simple slide and argue that the insights from Airedale, which we collectively call scientific wellness, fall into 5 general categories. So 1, there are 8 categories in the genome which deliver actionable possibilities, and the genome has tens of thousands of actionable possibilities unused. It's the most underused possibility for enormously improving health. And in a lot of cases, it's a 1 in 1,000 or a 1 in 10,000 case, but it can make an enormous difference in the individual. The second is we can take all of the data that we get from these analyses, and we can statistically ask which pairs of data in different information types are statistically related in a positive or a negative sense. And three, we're actually creating now physiologic trajectories that are completely distinct views of complexity, and each offers a multiplicity of actionable possibilities for optimizing the feature they are measuring. So biological age, BMI, the BMI is enormously flexible, adaptable, and totally comprehensible, and beautifully fits in with all the anti-GLP things that are going on these days, and fat control and so forth. Fragility, immune reactions, we can look at the relative interaction of the group in an active community. We're measuring trajectory for cognition, and because the brain is flexible apart from neuron loss, you can reverse deficiencies in cognition beautifully. And then finally, we have organ state changes that I already talked about for 25 different organs. For transition states, wellness, and disease, we can do early detection and think about clinical diagnosis, and I'll show you a couple of examples of this. And then finally, scientific wellness is really the complete encompassing bit for biological age and so forth. And if you had to ask, what is the one major outcome you'd like from all this scientific wellness, it's really quite simple. It's expanding your health span into the 90s, where you're mentally, actually, physically capable, and if you have a purpose, you can still get a lot done. And it's a terrific thing to think about in a time when all developed countries are way below the replacement birth rates. And that isn't going to turn around for a lot of really interesting psychological reasons. And the final thing I'd say is we have to integrate brain, body, and the gut microbiome. Those are the three major categories, functional kinds of categories. So let's talk about each of those. Here's just a list of the actionable possibilities from the genome. And the one that I'll talk about with regard to cancer that I think is enormously important are polygenic scores in assessing genetic risk. And there are really interesting things you can do with that. And I'll show you that in just a moment. Again, tens of thousands of possibilities come out of these things. The second thing is, in the first 108 people we looked at with 500 proteins and all the rest of the things, we found 3,500 statistical correlations, and they allowed us to map beautifully the paired correlations into networks that gave us insights into the biology of what they were speaking to. And I, for example, had really low vitamin D because I had two variants that blocked the uptake. And the only way around it was super doses of vitamin D, both to elevate my level and maintain things. Those are the kind of actionable possibilities you can think about. But another way you can use statistics, and is absolutely fascinating, is to take this incredibly tangled hairball of statistical correlations and use an algorithm called the community algorithm, which basically asks, what are the different classes of metabolites that are most closely related to one another by their statistical power and so forth? And to our amazement, when we did that, we found that 108 people, we could map out 70 different communities, and each of them mapped beautifully into either a normal biology or a disease pathology and so forth. And in fact, one of these, the cholesterol community, which we're expanding out now, had 12 correlations, LDL cholesterol, but other kinds of things. And one of the correlations was a really negative correlation with thyroxine. So we said, gee, here's really a good drug for LDL cholesterol. And Lilly was just finishing up a stage 3 clinical trial where they brought that drug out. And what we were then able to do is not look just at 500 proteins, but 5,000 proteins. And we got 10 times as many statistical correlations and hundreds of communities, and the communities fused heterogeneous data types together in ways we never had understood from the simple linear, one-dimensional biological networks that are typically analyzed. So, and we've gone on to do this with Airedale, and the results are spectacular. Let me show you what we can do with the metrics for physiology, and this is biological aging. So the key thing is we've been people from 21 to 93 into 10-year categories and looked at outliers for the 3 classes of analytes and saw in a pretty linear way as you aged, you lost control. And we could essentially use that as an algorithm for your biological age, and I won't go into the details of it. But what that algorithm did was, and the idea is the lower your biological age is than your chronologic age, the better your age, okay? And what the algorithm did was show for women in Airedale, they lost 1.5 years of biological age for every year they stayed in, and menopause 0.8 years. We were able to use the metrics to assess biological age of major organs, and they can be quite different from what your global biological age was. And we were able to use the features of the biological age to make all sorts of clinical suggestions on how to optimize your biological age. When we looked at people with disease, in every case, the biological age was elevated. And the really important thing is now we have the ability to take the features that make up these algorithms, and it's true of all the ones I showed you, and to feed them into a knowledge graph and to map them into networks and communities and put that all into a transformer so we can begin to integrate aging with scientific wellness, and I'll show you more about that in just a second. And then finally, the really important thing is because we chose people at all ranges of age, we were over a 4-year period able to construct the health trajectory of the human race with all the reservations you have that it isn't a single person. But that 4-year period is really important because we're going to use it to look at chronic disease later on in exactly the same way. And I'll show you about that. So, data-driven healthcare. In traditional medicine, of course, you have early onset of disease. It generally isn't very measured. It goes through a series of progressions up to the point where it can be clinically diagnosed. And as I said earlier, by that time, for almost all chronic diseases, you have a multiplicity of very complex biological networks that have been perturbed. And the best drugs hit symptoms. They don't hit the whole disease itself. What we did with Airedale was note that there were 167 wellness disease transitions, 35 in cancer. We analyzed 10 of those in the bloods that were drawn prior to the diagnosis. And we were able to show for all 10, we had a multiplicity of proteins elevated that occurred 1 to 4 years before clinical diagnosis, and 2, in many cases, mapped into classic disease-perturbed networks. And the importance of that is they tell us the beginning of the disease perturbation, so we can focus on that for reversing the disease. And that's exactly what we want to do with genomic medicine, be able to do very early diagnosis, pick out an appropriate target and reverse the cycle of these things and return us back to wellness. Okay, so that's one major approach to early diagnosis for chronic diseases. And let me just say, health has 3 stages, and data-driven health can optimize wellness. It can deal with early detection of transitions and inappropriate cases, hopefully reversal. And it can deal very powerfully with disease and all its complexity. But that leads to the idea most disease won't be dealt with by one drug. They're going to have to have a multiplicity of drugs. And that's a problem with the FDA, how it looks at clinical trials, and we'll return very briefly to that. A second approach are polygenic scores. We can convert, if you have a full genome sequence, any polygenic score into your genetic risk for the disease. And in 2019, we actually did a really interesting analysis that complemented this study that came out of the British Biobank in 2018, where the x-axis was increasing risk and the y-axis was disease feature for each of those 4 things. And what you saw is at the end of the highest risk, there's an exponential increase in the disease risk. So what we did is we replaced the y-axis by looking at all 800 analytes we had in the Airville population and seeing if there were any that were elevated or decreased. And in this 800 different analytes, for every single one of the 54 different polygenic scores traits we looked at, we found a multiplicity of analytes were either elevated and or decreased. And what this means is we could map out genetic risk for all of these things. And let me tell you today, for cancer, there are 251 polygenic scores. And there are a lot that need to be consolidated to make them much better than they are now. But it's an enormous resource for doing the kind of things I'm going to talk about next. And so let me talk about LDL cholesterol and proxy for heart disease and the polygenic score for increasing risk for LDL cholesterol is mapped out in the Airville population here with people that weren't on statins, okay? And what we saw is individuals at the highest risk, if they had high levels of LDL cholesterol, only chemistry could bring it down. If you were low risk, diet and exercise brought it down beautifully. So the really important point is, you want to know the polygenic scores of patients, because I'll guarantee you for most in the future, you'll treat high risk and low risk people quite differently. But a really critical question here is, when does the genetic risk manifest itself? Look, you have the genetic risk from birth, but you don't get these diseases until much later. And we fortunately had one individual in the 108 individual wellness study we did that had a group family history. 3 male members of this family had died in their early 50s from heart attack. So what he did was start taking his LDL cholesterol in his teens. And he found it was absolutely rock bottom level until mid-30s. And what was interesting is he was athletic, he was lean, he had a good diet. This is an intrinsic factor. I'm not saying it will always be, but the really key thing, if you're a doctor and you know this guy has a proclivity for that, what you can do is follow the corresponding analytes. And when they become elevated, you go to them and say, it's time for treatment. And I think we'll be able to do that for other kinds of chronic diseases. He's now 75 and living fine with this. I'm not saying it was the only thing, but it was certainly one big thing. Now, the organ-specific proteins for the brain and placenta, I give you 2 organs here. One, they map into really interesting biological networks. And two, in the brain, they map into geographical regions. And what is interesting is in the brain, which has more than 600 transcripts that are organ-specific, in the blood you see 10 or less at the level of sensitivity we're looking at. Until the brain has a disease, loses the blood-brain barrier, as we saw with scrapie disease, where these proteins poured out. And they, one, show you the biology, and two, they show you, in some cases, the location of the region for the blood-brain barrier. And again, we looked with this at acetaminophen in mice and humans, and were able, for the liver, where we had 84 organ-specific blood proteins, to beautifully map the onset of the activity and the homology between the human and mouse system. And we actually came up with 4 markers that were infinitely superior to the classic ones on the liver. But as far as I know, they didn't get translated to reality. So what scientific wellness is really all about is big data, as you can see all of these things, and it's discovery science. We're not creating hypotheses, we're looking at data and translating that data into what it tells us. And of course, the geroscience is quite the contrary. They focus on the hallmarks of aging. There are 14 of them now, and they are very focused and driven to understand each of those. And what we're doing, one of the reasons I thought for a part-time appointment, is I want to do an integration of scientific wellness and the features of the individual aging projects that are going on down there. And we'll do it by knowledge graphs and transformer integration and things like that. And I think we really have a great shot at moving, for most people, to a health span that's 90 weeks and beyond. I think the really interesting question is, what do you do with retirement? And what do you do with motivation? That is really important for aging. We each have to decide what those are about. So we can optimize wellness in aging. We can reverse the genome deficiencies or phenome deficiencies. We can actually utilize these novel physiologic trajectories to beautifully optimize wellness in the context of each of those different things. So data-driven health isn't just about healthy aging and preventing disease. It's about making you better. And that's really the important point. We obviously have the ability to do early detection and think about prevention in the future. We can deal effectively with complexities of diagnosed disease and really multimodal therapies. And that's what I was going to talk about with the new drug company, but we won't get time to talk about that today. We can identify biomarkers and get drug targets for wellness and prevention and disease, and have causal relationships that give us deep insights. So where do we want to go next? One, we're going to use bigger data. So we're going to generate 10 to 100 times more data than we did for Airedale. And it's all the classic things there. But I'm just going to give you a quick taste of the new technologies that we're exploring. So one, the polygenic scores we talked about and the power they have for disease reversal. Two, we're doing single cell analysis in select cohorts at each blood draw for 5,000 cells to define completely each cell type and its state of differentiation. And integratedly, that lets you determine spectacular results from that. We have a multispectral instrument that looks at 1,400 spectral lines across the narrow band of the wavelength. And our partner, Pattern Computing, has demonstrated in saliva, in 3 seconds, it can distinguish at 100% accuracy a normal patient from a COVID-19 patient. So we're mapping this into some of the things we're doing with the big ARPH that we'll come to in just a moment. Alpha structures are incredibly interesting in diagnosing neurodegenerative diseases. And I can explain all about what that is, but it's a totally new diagnostic approach. And there's a real hope of seeing things 10 to 15 years before clinical diagnosis. Brain health, especially cognition, we can measure and optimize. I have a friend at Stanford who's got absolutely a spectacular solid phase synthesis approach to creating millions of aptamers that you can use to assess their interaction with metabolites. And you can get exquisite specificity by virtue of the fact when you do that solid phase synthesis, he puts it together so if it interacts with something, it changes a color reaction that you can then take a photograph of a million things. So you can do a million analyses, you can do hundreds of metabolites and pour it through, and you can sort out which metabolites are uniquely specific with no cross-reactivity. And he's figured out how to use microfluidics and a cell phone to be able to look at a drop of blood and make 30 to 50 measurements and such a thing at all. So there are a lot of things that are going to democratize what we're talking about here. Voice analysis, Luxembourg has really done some beautiful work on this. 240,000 people with type 2 diabetes, absolutely fascinating stratifications and early induction changes in disease. And the one that I love is the hair analyses. So for every inch of your hair, they can measure 150 environmental contaminants. And if you have long hair, each inch is the equivalent to a year, so you can map out your history of environmental contaminants. And what they were able to do with this is from 14 centers in the four surrounding countries to Luxembourg, they were able to show they could geographically locate the person with regard to the contaminants they had. So this is, I think, going to be important for the future. We have a data platform to do omics and handle knowledge graphs and transformers that is compatible with all of the major clouds and so forth. And it's really designed for partners. We've actually transferred it to the Buck, and they've had really good luck with the whole thing. So what we're doing with regard to AI are two projects. One, we're putting the first level of actionable possibilities from all 5 of those categories into a knowledge graph. And then we're using that as data to educate a transformer that will be able to classify and then ultimately deliver to physicians actionable possibilities in the context of the patient's genome and phenome. And we'll come back to that. And we're using this to develop. We're looking at each individual and doing N of 1 analysis by measuring the variation of the individual over time from 4 different informational categories. And I won't go into those now, but it's going to work beautifully. And we'll be able to put that N of 1 analysis right into the actionable possibility analysis, and then have the ability to do both of these things. We now have a chatbot for patients and physicians into which we put all of the data on data-driven health, and it does a beautiful job of answering any question we can think about. And we've stripped out a lot of the hallucinations and things like that. So that obviously will replace coaches in the future. We have 2 major digital twins I won't have time to talk about. We're looking at 2 kinds of opportunities. One is the individual genome analysis of variable, and the second is the big data approach to the 4 major chronic diseases and then pregnancy and some other things. And with the big data approach, we can do early diagnosis, division into subtypes and their transitions, a match of the drugs and so forth. In this latter study, we don't deliver actual possibilities, we just get analytic data for doing things. The projects that are ongoing at 52 million grants over 4 years to look at rhythms in the blood, to look at the harmonization of very large genomic sets of data, and we have 3 sets, to take a big data approach to a 15-year study on type 2 diabetes that has blood draws from the clinical data. We'll make a multi-omic analysis. And then here, an impedance match of the ability of omics to assess things and mapping them into 2 wearables. Any wearable, if you look at the raw data, has an awful lot of information that no one knows what it's all about. And some of the wearables have enough information that we think we can really do this impedance match and move omics into the home if we can get wearables that can do what the omic does. We're having big projects in type 2 diabetes, starting with 1,000 individuals. It's just beginning now. We looked at pregnancy, 550 individuals, 5 times across pregnancy. We've already seen remarkable things in making predictions about maternal and fetal health and so forth. And then we just announced a global 1 million person project led by an individual in Turkey, focused in Asia. And he wants a 5-year program and maybe 10% return of actionable possibilities. And we'll talk about another one. These are the 4 major challenges, 5 major challenges of contemporary medicine: quality and aging and chronic disease, and equity and exponentially increasing costs. And I'd argue that the data-driven health really have powerful solutions for each of these. And of course, optimizing wellness and healthy aging, avoiding disease is a big step forward. But being able to deal with chronic diseases so that in a 10-year period, we can reverse a substantial fraction of the 86% of the $4.5 billion budget chronic diseases used today. And likewise, there was a study at NIH in 2015 that looked at the 10 top drug sales that showed on average 10% had a respective response. And we can map with data-driven health precisely the individuals that work with given drugs, and that's a big thing too. And then bringing the insights from the big data approach to chronic diseases and these kind of things. The million-person project, of course, is really key. I have just a couple more slides. Okay. It's going to let us through optimization of health, enormous reduction of healthcare costs, drive the technologies to the home, and generate the actionable possibilities we need in the future. So, the 3 things: 1, wellness and prevention and intertwined vaccine that we can all begin to think about practicing. 2, the democratization of healthcare to bring it into much broader context health. And then 3, the AI-physician partnership, which makes every physician a domain expert in much of medicine and hence able to treat the patients superbly. If you can get physicians to feel comfortable with their data. Okay, I'm done. Sorry.